Thank you everyone for taking the time to join this webinar. As host introduced, we will have a discussion about the antibody discovery strategy against multiple pass membrane targets. For starter, I will give a really brief overview regarding the current trending and the challenges in the field of antibody discovery against the multiple pass membrane protein. Then we will spend a little more time on the technical solutions regarding this particular application. At the end, we will share a case study of antibody discovery against the CCR8, a really popular immune modulating target over the last couple of years. So multiple pass membrane proteins are a class of antigens with a significant interest in biomedical research and drug discovery as potential targets for therapeutic interventions. Based on their functions, they can be classified into several families, such as G protein coupled receptors, ion channels, and transporters. Some of these targets, such as CCR8, CXCR4s, GPRC5Ds, are already in clinical trials or already launched in the market. So GPCR is one of the most popular family in drug discovery and development. However, the majority of these drugs, these launched drugs, 90%, 92% to be more specific, are small molecules. So far, there are only 2 antibody drugs launched in the market. Nevertheless, there are over 170 active pipelines targeting 76 GPCR proteins in the format of monoclonal antibody therapies, bispecific antibody therapies, ADC therapies, and gene and cell therapies. The main reason for the smaller number of GPCR antibody drugs lies in the challenges in antibody discovery. First of all, difficulty in antigen preparations. Multi-pass membrane proteins have sophisticated structures and are expressed at much lower level in the host cells. As a result, it is technically challenging to prepare the multi-pass membrane proteins with proper folding and biological functions. Given these features, the quality of antigen is less than ideal, leading to undesirable immune response and lower positive hit rate. So under these circumstances, GenScript ProBio has developed a tailored solution to tackle with these technical challenges. In general, we have established the novel antigen preparation strategies, including mRNA and VLP as the main immunization antigens, and generation of antigen overexpressing engineered cell line for binder and functional screening. In addition, an optimized hybridoma-based high-throughput screening approach known as the PowerDoma is established to facilitate the identification of functional candidates at earlier supernatant screening stage. With these platforms, we are able to get rid of the time-consuming subcloning process and obtain the candidate sequence within 2 weeks. Now, let's go over this process step by step, starting with antigen preparations. Peptide and recombinant proteins are the most popular antigen formats for immunizations. However, these two options are not really suitable for multiple pass membrane targets, mainly because they are unable to display the antigen's native conformations. Membrane proteins need the lipid bilayer to support its proper foldings. Without this bilayer, the epitope presented by artificial antigens is totally different from what it is in vivo. Currently, cell and DNA immunizations are the most common approach for multiple pass membrane targets. However, this combos here, the DNA plus the cell, has its intrinsic bottleneck that leads to undesirable immune response. DNA immunization can only generate a relatively lower level of antigen expression in vivos, resulting in weak immune response, while the cell immunization brings a significant portion of a non-specific immune response against those irrelevant antigen expressed on the cell. Unlike all of these approaches, the VLP and the mRNA are able to induce much stronger and much more specific immune response, leading to better hit rates during the early screening stage. In the next couple slides, we will have a detailed discussions about these two approaches, the VLP and the mRNA. First, let's start with the mRNA. The concept of mRNA immunization is pretty similar to the DNA immunizations. Instead of preparing artificial soluble antigens like peptide or recombinant proteins, we let the host animals to produce the antigen proteins in vivo by transfect the DNA or the mRNAs, basically the protein encoding gene sequences. Comparing to DNA, mRNA is able to produce the antigens with much higher abundance, leading to better immune response. In addition, the LNP package that brings the mRNA together can also serve as the adjuvant, which can further improve the efficiency of the immunizations. This is a case study of antibody discovery against one of the most popular GPCR targets, the CCR8. The purpose of this project is to compare the outcome of a traditional DNA plus cell immunization versus mRNA immunizations at 3 different stages. The first is in vitro antigen expression level measured by flow cytometry, as detected by 3 different reference antibodies. The mRNA, which is indicated as the blue bar here, demonstrated roughly 2-fold higher expression level on the CHO-K1 cells by transient transfection as compared to the DNA transfection, which is indicated as the red bar in this graph. After animal injections, the mRNA is able to induce much stronger immune response, roughly 30-fold higher than the DNA, as determined by serum flow cytometry analysis. Here in this graph, again, the blue bar represents the mRNA immunization, and the red bar represents the DNA plus cell immunizations. Moreover, this robust immune response enables us to shorten the immunization timeline by eliminating any further boost. As you can see from this chart, there is no forced immunization for the mRNA approach, while on the contrary, The DNA plus cell approaches requires force and further boost to improve the overall immune responses. Last and the most important, we are able to obtain much more antibody candidates, especially those with higher affinities during the screening stage. We compared the number of candidates recovered during the hybridoma screening stage and categorized them into different affinity range based on the fact MFI, the signal ratio. As you can tell from this table, the mRNA approach produces more candidates in the high affinity range comparing to the other two animals, which is taken with the DNA and cell immunizations. In addition to the CCR8 case studies, we did another one against the GPRC5D for the same purpose. Surprisingly, this case study gives even better results. The GPRC5D mRNA induces hundreds-fold of higher serum titers comparing to the DNA plus cell, as indicated in the male panels. And we recovered hundreds-fold more candidates in the higher affinity ranges, as indicated in these tables. With these two internal case studies and also a dozen completed projects after these two internal case studies, mRNA is becoming our top choice when it comes to immunization and antibody generation against multi-pass membrane targets. Besides mRNA, VLP is another powerful tool for immunizations. Similar to antigen overexpressing engineered cell lines, VLP approach also relies on mammalian cells to display the transmembrane antigens. However, it takes a step further by a process called budding. The VLP core will be encapsulated by the lipid biolayers and released from the cells, and the target antigen will be expressed and stabilized on the lipid biolayers. Each VLP can be considered as a mini version of the mammalian cells, but with much higher immunogenicity and much higher surface areas, leading to better immune response. The quality of a VLP can be determined by a standard ELISA assays with the antigen benchmark antibody. The data in this slide showed after 5 cycles of a freeze-thaw process, the GPRC5D VLP and the Claudin 6 VLPs samples still demonstrate robust binding curves and EC50s against the positive control antibodies against these two different targets. And in the case study of anti-Claudin 6 antibody discovery projects, we compared the immunization efficiencies as determined by serum flow cytometry analysis between the cell immunization group versus the VLP immunization group. As you can tell from these summary tables, the VLP induces significant antigen-specific signal shifts as measured by the Claudin 6 overexpression cell line versus the parental CHOK1 cell, while the cell immunization gives a pattern overlapping signals, suggesting that those antigen-specific antibodies are buried under tremendous amount of non-specific antibodies. After antigen preparation and immunizations, a powerful screening platforms can maximally preserve the antibody diversities and facilitate the recovery of desirable candidates. Traditional hybridoma technology is pretty well established and well recognized by researchers globally. However, the time-consuming process is still a major bottleneck for these technologies. Here at GenScript, we developed a optimized process called ProBio by plating the hybridoma monoclones in 384 well plates after fusion and high throughput sequencing right after the screenings. This workflow completely eliminates the subcloning process and reduce the timeline by almost 2 months. Moreover, the monoclonal state strategy at the very beginning facilitates the supernatant-based in vitro efficacy screenings, enable us to identify the functional candidates, the one with a biological function at a much earlier stage. And it can reduce the resources wasted on those binders, but not functional. So we're talking about the ProBio platforms. We actually did 2 case studies to compare the screening efficiency between the traditional hybridoma platforms versus the ProBio approach. In each of these cases, we started with equal amount of a plasma B cells between these 2 different approaches. Let me pull up the laser. And plate these hybridoma fused plasma B cells with different densities, which is 1 cell per well for the ProBio in the 384 well plates versus 3 to 6 cells per well in the traditional hybridoma approach in the 96 well plates. In both cases, the ProBio demonstrated roughly 2-fold higher positive hit rates. In addition, the ProBio process demonstrated robust features, including 97+% of a sequencing success rate, roughly 80% of a monoclonal rate, and 100% of a recombinant confirmation rate. So to put it in a simple word, the majority of those identified candidates from the supernatant screening step can be verified as true positive binders or functional candidates after purified recombinant antibody confirmations. Comparing the other 2 popular antibody discovery technologies, the failure displays and the single B cell screening, ProBio offers much more flexible screening strategies compatible with the various combination of screening schemes. More importantly, it enables better preservation of natural sequences, affinities, and developabilities. Now, with the ProBio upgrades approach, it significantly shortened the overall timeline from 4 to 6 months to 3 to 4 months, makes it comparable to the timelines of the other two platforms. As I mentioned at the very beginning, the in vitro function assays is also a crucial part for the discovery process. We have established the high throughput efficacy screening assays covering more than 130 GPCR targets for the identification of agonists, antagonists, and allosteric modulations. In addition to the in vitro pharmacology, ProBio team also provides a comprehensive in vivo animal work, including PK, PD, and non-clinical safety studies from discovery to preclinical development stage. At the last part, I would like to share an internal case study of antibody discovery against the CCR8 using the technologies that we have just discussed previously. CCR8 is a pretty popular immune modulating target in recent years. It is predominantly expressed on Treg cells in tumor microenvironment. The working theory of the CCR8 therapeutic hypothesis is targeting the CCR8 to inhibit and to deplete tumor-resident Treg cells. This is an overview of the screening funnels for this case studies. We started with mouse immunization by DNA, cell immunization, and mRNA, followed by hybridoma fusion and screenings. Those hybridoma candidates were screened by high throughput flow effects analysis against human and cyno CCR8 overexpressing stable cell line, and further validated by ADCC and Wilson flux assays for functional efficacy. The top candidates were humanized and confirmed by in vitro assays, efficacies, and developability assays to narrow down to the top two leads for the subsequent cell line generation and process development. During the early screening stage, we obtained two panels of candidates generated from either DNA and cell immunization or the mRNA immunizations. These candidates were further evaluated by calcium flux assays and ADCC assays. These top three candidates demonstrated better in vitro efficacies in both assays as compared to two clinical benchmarks, the one from Gilead and the one from BMS. Based on these data, top two candidates were selected for humanizations and the reconfirmation of the in vitro efficacies by ADCC and the calcium flux assays in comparison with these two clinical benchmarks. Besides functionalities, binding affinities towards the human and cyno CCR8 cell lines were conducted by FACS. And these two humanized candidates demonstrated much better cross-binding affinities to cyno CCR8. In addition, we also looked into the potential of these two candidates as ADC drugs by antibody internalizations. IncuCyte was used to capture the kinetics of the antibody internalizations by CCR8 overexpressing engineered cell lines. These two candidates, as indicated as the green ones, show much better internalization potentials comparing to those two clinical benchmarks. Although they share pretty close or similar epitope as determined by the epitope competition assays on the right panels. At last, we carefully evaluated the developability of these two candidates with a panel of biophysical studies, including thermal stabilities, aggregations, SEC, and CSDS purities. Both panels gave desirable performance, suggesting that these tools are very viable candidates for cell line generation and process development. Okay, so that's the end of my presentations. Let's move on to the Q&A session.